Hello, everyone, and welcome to today's webinar, Determination of Cell Surface Markers on Circulating Immune Cells as Biomarkers for Disease Monitoring Using Spectral Flow Cytometry. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Cytech Biosciences. To learn more, visit cytechbio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Aymeric Silvin, Research Immunologist at the Gustave Roussy Institute. Dr. Silvin, you may now begin your presentation.
Thanks very much. Thanks to give me the opportunity to speak about my work. So today I'm going to speak about two different stories. The first one is about COVID. So this story happened in the midst of COVID in March 2020. And it was a big collaboration between Gustave Roussy Institute, between Eric Solari and me, but also between Paris Hospital, Michaela Fontenay. And what happened is that, as you all know, this disease spread quite very fast. And as a Paris Hospital were completely overloaded with patients, and ICU were in a critical stage where they had to choose who they needed to put in ICU and who had the highest chance to be saved. So the question was how to identify severe COVID patients before that they enter in the severe phase. So to assess this question, we used blood samples, and we harvested blood from this patient and their plasma, but also we put some of the blood into a cytodelics solution that allowed to conserve all the immune cells and to study neutrophils. And we also perform RBC lysis in order to perform single cell. So here you have the different techniques that we use. We use single-cell RNA sequencing. We use spectral cytometry. We used CyTOF to make sure that the spectral cytometry data were concording with the CyTOF, since it was a new technology that we acquired in the lab. And we finally performed cytokine and chemokine quantification using the MSD technology. So here, for example, what happened from the blood, I elaborated a panel of 25 markers that would identify each immune cell population present in the blood using the spectral cytometer Aurora from Cytech. So this analysis, we decided to perform a non-supervised analysis in order to extract all the information in the fastest manner. I have to precise that this story took 4 months to be published. So basically the article was finished in 2 months, and then we had 2 months of revision. So as you can see here, in this UMAP representation of spectral cytometry data, we were able to identify the neutrophils that express CD15, CD66B, as you can see on the panel C, bottom part. We were able to identify the monocytes that express CD45 high, but also CD14 and CD11B. We were able to identify the B cells that express CD19, the T cells and the TCD4 T cells and the TCD8 T cells, and the NK cells. So from this identification, based on the regular markers, so that were part of the panel, we then checked if we could see a change between the 3 categories of patients that we had. We had a category of patients that were called control, meaning that they arrived at the hospital with flu symptoms, but they were tested negative for COVID. Then we had a second class of patients that were tested positive for COVID, but not displaying any breathing difficulties. So we considered them being mild. They had fever, they had muscle pain, but no difficulty to breathe. And finally, patients that were presenting severe COVID, who were presenting a certain amount of lack of oxygen in their blood, and were displaying difficulty to breathe. And when we compared the immune cells using this 25 markers panel, we observed that the main population who was changing in these patients were the neutrophil, as you can see in the panel D, the bottom part. You can see that the neutrophils from control patient are slightly different from the mild COVID patient, and the neutrophil from the severe COVID patient are very different from the mild COVID patient. So then, of course, we came back to a regular analysis of the cytometry data, and we observed that there was a higher percentage of neutrophil in the blood of COVID, severe COVID patients compared to the mild versus the control. But on top of that, we observed that the neutrophils in this severe COVID patient were losing two markers, CD10 and CD101, as you can see here when we represent in a regular cytometry two-parameter visualization. And this coincide, you can see here the quantification of this CD10 low, CD101 low neutrophils in severe patients. So we decided to explore their signature and their single cell signature. So for that, we had to face the fact that neutrophils are not really well endowed in order to perform single cell. And at this time, Gustave Rossi was acquired 10x 10x technology. So what we did is that we did a quick RBC lysis in order to remove the red blood cell from the blood of the patient and perform a 10x single cell RNA sequencing. So this was very challenging. Why? Because we needed to have fresh blood from patient with a mild versus a severe COVID and some control. And the hardest was a mild patient because the patient had to come to the hospital, but without having severe COVID, and we had to ask the patient to come back 10 days later to withdraw again blood from this patient. So without any question, what happened is that this is the blood from a nurse, so a hospital worker who presented COVID, a mild COVID, and who accepted to have their blood taken again 10 days later. And what you can see here is that neutrophils from this patient, mild COVID patient, display a very strong interferon signature at D0 of the symptoms, and 10 days later, this interferon signature has been completely downregulated. So it seems that mild COVID patient has a transient interferon signature, so interferon induced signature probably, and then it disappears very quickly. However, in the severe patients, we were able to observe completely different signature, and among the DEGs, we were able to see DEGs MME, CXCR4, who were suggesting that these neutrophils could be immature. And another DEG was S100A9 and S100A8, who are two proteins that, when they are assembled, form calprotectin. And what was really striking is that every morning we would gather with the medical doctor from the hospital, researchers, and we would discuss what we were saying, us, at the immune level, using our technology and in the research laboratory, and the medical doctors were telling us what they were seeing happening in the patients. So it's from this double discussion between researcher and medical doctors that we started, of course, to speak of the cytokine storm, but it allowed us to create hypotheses and to try to understand what was below the cytokine storm. Because of course IL-6, TNF, IL-1 beta were guessed to be very important. However, when they were quantifying these cytokines, it was not as striking. And for example, we decided to use the plasma of this patient to quantify 42 cytokines and chemokines. And we used two different techniques, and in total 84 patients. So of course we didn't do single cell RNA sequencing on a lot of patients, but the goal was to validate this data at the protein level in a lot of patients. And here you can see the comparison between control patients versus mild COVID patients, and the main cytokine that was found in mild COVID patient was interferon alpha, while in the control it was completely different cytokines and not interferon alpha. And when we compared the control plasma versus the severe COVID patient, the cytokine that was the most produced differentially in the severe COVID patient was calprotectin. So the assembly of S100A8, S100A9. And we quantified other markers, other cytokines. And here you can see in the bottom part, CXCL8 production, which is at a regular level in control. It goes down in the mild COVID patients and it goes up in severe COVID patients. You can see that it goes up, but it's significant, but it's not completely. You do have severe patients that produce the same amount of CXCL8 than the mild patient, than the control patient. So CXCL8 was not the best to follow and to categorize patients. IL-6 was similar, meaning that there was an increase of IL-6 in the plasma of severe COVID patient, but it was not as high, and it was not discriminating completely severe patient from mild patient or control patient. Same for interferon alpha. You could see an increase in the mild patient, but you could see also some interferon alpha produced in severe COVID patients. So finally, we quantified calprotectin, and here what was really interesting is that you had almost no overlap between the severe COVID patient compared to the control, and you had an overlap. Some of the mild patients were producing as much as calprotectin as the severe COVID patient, but there was a group of mild patients that was not at all producing similar amount of calprotectin, and it was very similar to the control level. So I will come back to that later. Then we wanted to validate the fact that these neutrophils were immature, and we wanted to make sure that to compare spectral cytometry with CyTOF in order to have another technology to make sure that the results that we were seeing using our spectral cytometer was reproducible. And here you can see on the top part CD10, CD101 expression on neutrophils in a cohort of more than 60 patients. And you can see that indeed in severe COVID patients, neutrophils lose the expression of CD10 and CD101 on their surface, and this correlates with an increase of expression of CXCR4, which is a marker of immaturity at the level of neutrophils. Then we checked by CyTOF, and you can see that we saw similar trend meaning the loss of expression of CD10 and CD101 on the surface of neutrophils in severe COVID patients. So this work was performed with Eric Solari. Dr. Solari is a medical doctor who is specialized in a certain type of leukemia, and is expert in monocyte and the non-classical monocyte to be used as a diagnostic tool for a specific type of leukemia. And here, for example, he wanted also to check the monocytes and if there was change at the level of the monocyte. And here again, when we look at the monocyte by single cell of this patient, we were able to see that in the patient presenting mild COVID, there was an interferon signature, and this interferon signature came back to normal 10 days later. However, in the severe COVID patients, there was a specific signature associated with the monocytes, and it was the expression of CD141 and the expression of CXCL8 or NF-kappa B, IA or IZ. So suggesting that there was a chronic inflammation happening in this patient at the systemic level. So when we look by cytometry, we realize that indeed monocytes were also affected. And if you look at the panel G below, you can see that in a control patient, the patient display monocytes that express CD16 alone and CD14 low. These are the non-classical monocytes in the green box. And then you have the classical monocyte CD14 high, CD16 low, which is in the red box, red gate. And when we move to the mild patient, we don't see any drastic change between the proportion of non-classical versus classical monocytes. You see a bit of increase in the intermediate monocytes. And when we move at the severe patient cohort, you can see that in early severity, there is a loss of the non-classical monocytes, and after 8 days in ICU, these non-classical monocytes come back. And this is it is quantified in panel H and panel I. You can clearly see in the panel I, the non-classical monocyte dropping completely in early severe COVID patients and coming back after 8 days spent in ICU in the blood of these patients. We quantified CD169 on the surface of the classical monocyte. Why CD169? It's because it's an interferon-induced gene. And you can see that this is drastically increased at the surface of the classical monocyte in the mild patients, and not so much in severe COVID patients. You still had some patients that express a bit, quite a lot in the severe. So it was not that black and white. And CD141, who was among the genes in the previous slide detected by single cell THBD gene, was overexpressed in severe COVID patient, and severe COVID patients that were expressing higher level of CD141 on the surface of the classical monocytes were the one who spent more than 8 days in ICU. So all that to show that through spectral cytometry, we were able to, by using just a 25 marker panel, to identify key immune population that seems to correlate with the severity of COVID. So the question was, can we use this population as a predictive marker from a patient who is moving from mild to severe COVID? And for that, what we did is that, in fact, in our cohort, we had moderate patients, meaning patients who required oxygen bottle, but they didn't require yet ICU. And when we studied this patient, we observed that indeed, in this patient, we had an increase of neutrophils losing CD10 and CD101. And when we look at the calprotectin production in the plasma, this patient was intermediate between completely mild patient who didn't require any oxygen, and severe patient who required ICU. So suggesting that calprotectin could be indeed a great tool to predict COVID severity and to predict if a patient is moving towards COVID, severe COVID. So one of the questions of the reviewer was, what is happening in the lung of the patient? Can you see this population in the lung of patients with severe COVID? So of course, like at this time, it was very complex to have bronchoalveolar lavage from patients in French hospital. However, there was a paper who was published who did a single cell in the of immune cells in the blood and in the bronchoalveolar lavage. And here you can see the cells. We were able to identify the neutrophils. And when we look at the blood of control versus mild versus severe, you detect neutrophils. And when we look at the lung, you can see that there is almost no neutrophils in the lung of control patient. There is a bit of neutrophils in the lung of mild COVID patient, and there is a lot of neutrophils in the lung of severe COVID patient. When we look at the signature of these neutrophils, we observed that these neutrophils in the lung, in fact, were indeed overexpressing S100A8 and S100A9 that, when translated in protein, produce the calprotectin, and they were also overexpressing CXCR4, who is a sign of immaturity. So as you can see here, this was reproduced in the tissue of COVID, in the lung of COVID patients. So then, and this is the part where I have to say it was really great to collaborate with medical doctors, because I'm going to show you a graph that is very hard for me to understand, really, like the mathematics and everything behind. But through this collaboration with medical doctors, they were able to run a ROC analysis, determining which parameter was the best to predict COVID severity in patients. And among the different parts, the interferon versus the non-classical monocytes versus the CD16 low expression and versus the calprotectin. As you can see, the calprotectin was the one parameter who showed the best prognostic capacity. So this study ends there. However, then Eric Solari and Michaela Fontenay, Dr. Solari and Dr. Fontenay, decided to continue and to check in a bigger cohort of patients if calprotectin could be used really as a prognostic marker. And they published this article, Dynamic of Circulating Calprotectin Accurately Predicts the Outcome of Moderate COVID-19 Patients. And they published this tool where now medical doctors can enter the age of the patient, the height, the weight, the sex, and then the amount of calprotectin which is quantified in the plasma, if it's a EDTA plasma citrate, or if it's a serum. And with two concentration of calprotectin, there will be able, the algorithm will be able to predict if the patient is moving towards severe COVID. So the conclusion of the full article is represented here. In mild COVID patient, indeed, you have a antiviral response, which is mostly type 1 interferon, and this allows a maintenance of proper hematopoiesis and myelopoiesis with proper monocyte, classical monocyte, mature neutrophils, and non-classical monocytes circulating in the bloodstream of the patient. And in severe COVID patient, what we think happens is that there is a cytokine storm, but below this cytokine storm, there is a calprotectin, who is a very good marker, who increase with time before that the cytokine storm starts, and can help to predict which patient is moving toward the cytokine storm. And this contributes to emergency myelopoiesis with the appearance in the bloodstream of immature neutrophils and the decrease of non-classical monocyte. This decrease of non-classical monocyte in the bloodstream could be due to the fact that they infiltrate the lung. So it would be just like a localization. And that's why it would be very transient. So, of course, this study was, you cannot publish an article in 4 months alone. So it was a massive work effort from the Food Institute, Paris Hospital, and many collaborators. And now I'm going to move to the smaller study, but a more recent one. And we still not published. And this study concerns a disease which is called giant cell arteritis. And what is very strange is that it's a disease of the temporal artery. It happens usually in adults that are over 50 years old. It's an inflammatory disease. It's part of a common disease called vasculitis. It's inflammation of the blood vessel. And the symptoms are fever, weight loss, night sweat, asthenia, headache, jaw claudication, scalp tenderness, but the most important is the visual loss. And this visual loss will not be recovered. What is the diagnosis? The diagnosis is that the wall of the artery is gonna be inflamed, and as you can see here, there is a wall swelling and of course a decrease of the bloodstream. So this disease, which is called GCA, is diagnosed through the removal of the temporal artery and the analysis by an anapath of the thickness of the wall and the presence of giant cells in this inflamed wall. So, as you can guess, removing a temporal artery is not a tiny thing, and if the medical doctors are unable to diagnose the disease with the removal of one artery, they cannot remove the second one, otherwise the patient will lose their scalp. What do we know? It's an inflammatory disease. It has a very similar pattern with the PMR, polymyalgia rheumatica. However, it is not fully affecting the same part of the body. GCA affects the head, when polymyalgia rheumatica affects other part of the body. And the way of treating this disease is mostly using corticosteroid in order to decrease the inflammatory response. But one of the questions is, are these two diseases linked or related? Because some patients who have GCA have also PMR. What do we know about the immune response? What do we know is about T cells, which was very strange for me because it's an inflammatory disease and there was nothing published about innate immune cells. So that's why I found it very interesting for us to study. So what happened is that we had a cohort of 20 patients. We built a panel of 22 markers just to characterize immune cells. And using this panel, you can see here the markers that we use. Using this panel, we checked which immune population was drastically changed in this patient. And we use cytolytics again to preserve the immune cells and to be able to analyze neutrophils. And here, as you can see, is that when we compare GCA patient versus non-GCA patient, the massive difference is the neutrophils again. So we decided to look more carefully at the phenotype of these neutrophils. So here you can see like the fact that these neutrophils seems to overexpress CD68 on their surface in GCA and in GCA patient. And what happened is that we created a new panel which was more functionalizing panel, so which was a bit bigger, and the goal was to really characterize these neutrophils and to determine which activities they were presenting. And for that, it's what we zoomed inside the neutrophil. So here it's only neutrophils. And if we look in an unsupervised manner, we were able to split from control patient, PMR patient, PMR plus GCA patient, and GCA alone. And what was really drastic is that if you look here, controls versus GCA, you have a drastic increase of this type of neutrophils located in the red gate in GCA patient. And this is completely abrogated when the patient has been put under corticosteroid. So we decided to check what was the markers driving this clear non-supervised clustering of these cells. And we found that CD68 overexpression and CD62L overexpression were drastically increased on the neutrophils in GCA patient. What is very interesting is that CD62L is selectin, allowing the neutrophils to roll on the vascular endothelium before to perform diapedesis. And in order for the neutrophils to enter in the tissue, CD62L need to be cleaved, and then the neutrophils can perform the diapedesis. CD68 is a receptor for oxidized LDL, and this we still need to study it in more carefully. So here you can see that indeed in GCA patients you have a drastic increase of these two markers on the surface of the neutrophils. And then the goal was to come back because, spectral cytometry is great to perform big panels, to characterize things and to find markers. But however, at the level of the hospital, what medical doctors need is a very easy tool that can be used everywhere on the most simple machine. And so in order to perform that, what we did is that we came back to a very basic panel of 5 markers that could be used on any cytometer. And for that, we use CD14, who allows to check more like monocytes, CD15 and CD66B to characterize neutrophils, and then the 2 markers that were overexpressed in GCA patient, CD68 and CD62L. And using this very simple panel, we characterized more than 100 patients, and then it was the work of a wonderful statistician, Oriane, who did this massive analysis of 100 patients and who determined statistically if it was significant. And as you can see, in control patients, neutrophils do not co-express CD68 and CD62L. In GCE patients, you can see population of neutrophils co-expressing these 2 markers. In other inflammatory patients displaying all other type of vascularitis, of autoimmune disease, of other inflammatory disease, you see that there was no neutrophils co-expressing CD68 and CD62L. And most interestingly, when the GCA patient were displaying neutrophils co-expressing CD68 and CD62L were put under corticosteroid and tested again later, they were able to see that this population of neutrophils was completely abrogated. And this was, of course, significant here. And you can see the decrease in the GC remission. And finally, Oriane performed a ROC curve, and you can see here that it seems that the co-expression of these 2 markers on the surface of neutrophils is display a very good sensitivity and specificity to diagnose GCA. So the goal now is, of course, to publish this data, but to try in a multicentric manner in different hospitals around Paris to try this panel of 5 antibody in order to diagnose this disease in an easier manner without removing the temporal artery. So this work is really like also a collaborative work. It was between the Necker Hospital with Julien Rossignol, the Saint-Antoine Hospital in Paris, Gustave Roussy, Montfermeil Hospital, Curie Institute with Oriane de Beaupuy, who was the statistician who performed all the tests, and Melun Hospital. So I really want to emphasize that everyone is bringing something to the table, and doctors are bringing their expertise. They are bringing like a systemic and a full organism knowledge, and researchers are bringing their expertise, technical expertise, but also strong expertise in the immune cells that they study, and I am specialized in myeloid cells. And it's through this permanent communication between all of us that we were able to really like create new tools that could be used to change like patient diagnosis, and also to determine the prognostic value of a marker in the case of COVID. This here you can see the full team. We are even more now. We are 45 people. We are located in Gustave Roussy. So, of course, we work also on cancer, and we work also on neurodegenerative disease. And this is all the studies that I didn't speak during this presentation. But of course, we use spectral cytometry for these different other projects. And if you are looking for a postdoc, if you are looking for a PhD, don't hesitate to contact me or Florent Ginoux. We are always recruiting, and we are always looking for a young, innovative mind. Thanks a lot. And now I think we have time for questions.
Yes, thank you for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's get started. Our first question asks, what is the big advantage of using spectral cytometry compared to regular cytometry?
So in my case, what happened is that the big advantage I saw is that, of course, if I use the same amount of marker, for example, a panel of 12 markers, let's say that before I would spend like a half a day compensating this 12 color panel. Now with the Cytec, I would speak like not even an hour to compensate 12 parameter. But you can, so most of the time what happens is that you complexify and you do a bigger panel, but for a 25 marker, I will spend a half a day, when before I was spending the same amount of time for 2 times less marker. So there is a gain of time, and with Cytec Aurora, the massive plus is the autofluorescence. So you have a removal of the autofluorescence and a higher sensitivity for some markers, because you remove the autofluorescence of your cells, when before with regular cytometry you could not remove the autofluorescence of the cells. So I would say that the big advantages are these two, like a gain of time in terms of analysis, and the acquisition of a new parameter, which is the autofluorescence. And of course, like if the autofluorescence is uniform between your cells, it's easy. If it's different between your cell types, then you have to be more like it's a more complex manner, but you can still do it.
Wonderful, thank you. Okay, moving along here, next question. This is asking, do you face issues with your panels or analysis?
So yes, we always face issues, like it's trial and error. and because I'm very stubborn and I didn't know that I could get help from Cytech. So you do have people that are specialized in Cytech to help you and guide you how to design your panel. However, me, what happened is that I had to design them during the COVID period. I didn't have the contact, and it was like kind of troubled period. So what happened is that I designed them by myself, but it's like it's quite good sense. Like it's basically you don't want to put two markers that are co-expressed by the same population in two channels that are very close, because you know that otherwise you're gonna have massive compensation issue. So you follow these rules, and also like sometimes like if you really want to do a complex panel of 40 markers, you definitely need the help of Cytech to design this complex panel, but you don't always need 40 markers. And I do remind that you can use the Spectral for 12 markers or 6 markers, and it's a gain of time, and it's okay. Like it's fine. It's a cytometer.
Wonderful. Thank you. All right, next question here. This question asks, how did spectral cytometry change the way you answer your scientific questions?
So I think it's where, like for me, before regular cytometry was to validate something. Like you had a single cell and you validate it at the cytometry level. Now we can really use it. We can really like use it as a tool to see what is changing, and then go to characterize it, functionalize the cells using panel of functionalization panel, which was like not that easy. Before you had to choose, like you had to choose if you want to see all cell population, or like here you can really do a bit of functionalization, like a bit of characterization. I think like it really, yeah, I showed two studies that are not built the same way to address two different kind of question, but at the end we use the same technology. So yeah, it's very exciting, and you have much more opportunities, so you can use the technology in different manner. So it changed the way you address your question.
Excellent. Thank you. We have some great questions coming in, so please make sure you submit questions into the Q&A inbox in the lower left corner of that screen. Our next question that we have here is asking, do you have experience with markers identified by flow in patients with psoriasis, or more specifically, psoriatic arthritis before and after biologic therapy?
So for psoriasis, I'm not sure we have, but for psoriatic arthritis, yes. We for arthritis we tried and we saw, and it is not co-expressed. But it's not CD62L and CD68 is not co-expressed by the neutrophils in arthritis patient. So clearly, it seems that it was very specific to GCA, which is very like I really want to perform single cell on the immune cells to try to decipherate which pathway are upregulated and what could be the reason. Is it infection? Is it microbiota dysbiosis that could induce the systemic reason, and why the temporal arteritis? Like while the temporal artery, like this, I don't know if it's just a physics question, like these neutrophils are activated, sent to the circulation towards the temporal arteries, and get stuck there because there is an angle and stuff. So I really don't know, but I want to characterize it better. Definitely.
Wonderful. Thank you. All right, we have another question in here. This next question asks, what other markers can we analyze to ensure results about the severity of COVID-19?
So now, like there was a bunch of other markers published, and we are not the only one. There was C5AR1 also. There is a lot of other markers, but what for me is very interesting with calprotectin is that there was many papers after the COVID period who started to report it, calprotectin, as a very good prognostic marker for many inflammatory disease, like even lung adversary influenza. When patient with influenza start to do adversary events at the level of the lung, there is like there is a new, like there is a very good review that was published very recently, like this month, about how calprotectin could be a surrogate to CRP, the C reactive protein, which is used for inflammation in general. And in fact, calprotectin could be as good as CRP and even better. So I think like it's just to show that even in the 21st century, we don't understand everything, and the role of calprotectin is not fully understood. We don't understand really how it acts on the bone marrow. Is it acting on the bone marrow and the myelopoiesis? Is it contributing to the emergence of immune cells that will contribute to an inflammatory symptoms, systemic symptoms? Like there is many questions, and me like it's during this period I really felt like a detective, because I had medical doctors telling me what was happening in the patient. I had my scientific knowledge trying to compute what I was seeing with my data and how we could link everything together. And it's how calprotectin emerged, because I was convinced that inflammatory cytokines IL-6, IL-1, and TNF alpha were the last wave, the final wave, and that there was other stuff coming before who would contribute to fuel this IL-6, IL-1, TNF. So yes, but there is like plenty of them. The complement system, there is like really like it's where COVID helped to make emerge a new re-emerge stuff that were already spoke about before but forgotten.
Wonderful, thank you. Okay, and we had another question come in. This question is saying, your data on mild and severe COVID is impressive. Do you have similar data regarding patients with so-called long COVID?
So this is a very interesting question. Like sadly, we didn't go for long COVID, but I do think it's very interesting, because I do think that this long COVID patient do have a inflammation which is persisting. Is it at the level of the central nervous system? And I do think like it's a very crucial question, because we need to check if there is inflammatory cytokines that are continuously produced, maybe in the central nervous system, and all the central nervous system stay in a state of inflammation, even if there is no more inflammation. So I am convinced that long COVID is a persistence of the organism responding to an inflammatory response, and that's why patients like experience this exhaustion, like very hard time to concentrate, very exhaustion. So yes, it would be very interesting to have the plasma of this patient, but sadly no. I completely stopped after the COVID, because I'm in a cancer institute and I need to focus on cancer.
Absolutely. All right, and it looks like that is all of the questions that we have for today. I want to thank you again for your time today and your important research. We would also like to thank LabRoots and our sponsors, SciTech Biosciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thanks a lot. Bye-bye.